So as I introduced earlier, we classify the 20 amino acids into 4 different classes. And the first is going to be the nonpolar amino acids, of which we count 9. That means this entire page is all the nonpolar amino acids. Now let me make sure you understand something right away. As you look at the core structure of an amino acid, I would hope you would say, that is extremely polar. After all, it's got polar bonds, polar functional groups. It even has full charges. We said that's super polar, and it's true. All amino acids are polar. They're all highly water soluble. However, when we say that an amino acid is nonpolar, we're talking about the variable group. And that is going to be what we want to fill in here. You can see we have a lot of amino acid core structures here, and they all have essentially the same zwitterionic core structure with the negative and the positive charge. At this point, we're going to simply fill in the variable group, which we'll sometimes call the side chain, because it hangs off the side in this case. We're going to start with the simplest and smallest side chain, that of glycine. I'd like you to know the word glycine, its structure, and the 3-letter code GLY. And the variable group, which is attached directly to the alpha carbon in the center right here, is a simple hydrogen. It's as small as we can get. And what that means is that glycine is known as the smallest amino acid. And you might recognize that immediately we go back on what I said earlier about amino acids, which are chiral. This one is achiral. It's the only one of the 20 amino acids that doesn't have a D and an L form because it doesn't have 4 different groups around the alpha carbon. Lastly, we're going to say this strange thing, and I'll explain it a little more later. This glycine is going to contribute the most flexibility to a protein chain when it is incorporated into a protein. And it's not obvious why that is, but we'll see it later. So glycine is really known for being small and flexible, or adding flexibility to the chain. It's a real outlier, and it's an important one, but we'll leave it at that for now. Moving on, we want to talk about the next smallest, that is alanine, or ALA. That has a variable group that is simply a methyl group. It's nonpolar, it's inert, but we're going to say that it's not the smallest, but we'll say small, non-reactive, obviously nonpolar because that's the page we're in right here. So before we move on, I just want to point out that all amino acids have an alpha carbon. The next carbon out, in this case, is going to be called a beta carbon. And glycine doesn't have a beta carbon, but alanine and all the other amino acids do. So, great, let's move on. The next 3 down here, we treat them all as a group. It's going to make it easier to remember them. They're called the branched aliphatic amino acids, and we treat them, like I said, as a group. Valine has a beta carbon, just like alanine does, but it also has 2 carbons attached to that. I'm going to switch to stick structure from saying full out formula, just so that we can draw a little quicker. Valine looks a little bit like a V if you turn it on its side, so that'll work for us. VAL, valine, is slightly larger than alanine. Leucine has a beta carbon and then a gamma carbon. Gamma looks like that. And then 2 branched methyl groups coming off of it. Isoleucine, like valine, has a branch at the beta carbon, and then one of those extra carbons has another carbon coming off it like that. So all of these 3 together are medium size, and of course nonpolar, and therefore non-reactive. So you're going to treat these much as the same, and we'll talk about later, if they're so similar, why do we have 3 different ones that look so similar. Notice that leucine and isoleucine are isomers of each other. That's where the name comes from. And that is worth a note there because they're so similar in size. Moving on, we're going to go to the next side of the page here. We've got some unusual ones. We have methionine, which is MET. That is essentially a long floppy chain, and we'll make a note of that. And what we can see here is we've got an alpha carbon and then a beta carbon, another carbon, and then a sulfur, and then another carbon. That sulfur is not very reactive. Therefore, we treat it almost as if it's just 4 carbons in a row. That's pretty much all we need to know about it, except one biological fact, and that is it's always first in a protein. Just like a train always starts with a locomotive, the methionine is always going to be first in a synthesized protein. Let's skip proline for a moment and move on to phenylalanine, PHE. Phenylalanine's name serves it pretty well. We have an alanine right here with its carbon coming right off the alpha carbon, and then we have a phenyl group out here. So these two last ones that we're interested in down here, phenylalanine and tryptophan, are all very similar. They're bulky and they're aromatic, and of course they are nonpolar. So these are going to be the largest nonpolar amino acids, and they're flat and aromatic. They're not terribly reactive. Phenylalanine is not reactive at all. Tryptophan, TRP, has a hydrogen bond donor. Despite the fact that it's overall very nonpolar, it does have the ability to act as a nonpolar group. I want to emphasize to you one thing too. We tend to focus on the ring structures here. Phenylalanine's ring structure is easy, tryptophan's is hard, but don't forget these both have a beta carbon here, and sometimes students leave those out when they draw structures. So those are difficult to draw, or at least tryptophan is, and they're large, but nonetheless they're important and we need to know them. We're going to finish this page off with the last and most unusual amino acid, proline. Proline is a slightly different core structure. We start with an alpha carbon, we go to a beta carbon, and then we go to a gamma carbon and then a delta carbon. I'll put that delta down there, and that looks like a little D. But the interesting thing about proline, PRO, is that the delta carbon actually rings back and forms a single bond with the core nitrogen amine. Now remember, this should be protonated, and it is, and it has a positive charge on it. It's still zwitterionic, but instead of having an NH3, it only has an NH2 because that nitrogen has 2 carbon bonds and 2 hydrogen bonds. So it's really all about the number of bonds. It has 4 bonds, that makes it positive. Proline's a tough one. It's going to be very unusual to draw, especially when we become part of a protein. One last thing before we leave this proline, it is medium size and nonpolar, and it is the least flexible. It's kind of the anti-glycine. We said glycine is the most flexible when it's part of a protein. Proline, with its rigid ring structure connecting back to that nitrogen, makes it very hard, actually impossible, to rotate around this bond right here, and that makes it the least flexible. So more on that coming in the future. That's the end of our nonpolar amino acids.